I'm James Pearce. I'm a PhD candidate at Exeter University, and my project is looking at the early development and genetic engineering of the waxworm moth Galeria melanella. So I was looking at PhDs, and it's pretty much the same as looking for a job. You look at certain things. If the description sounds interesting, then you might apply for it. I saw this project, and it was a combination of working with microbiologists who are the primary users of wax moths as a model organism, but also it was the opportunity to take work that's been done in fruit flies in Drosophila, where they have a very good understanding of how cell division works in that particular insect, and to look in moths, which actually hasn't really been done. There was some work done back in the 1980s, but really it was sort of been left there because Drosophila was an easier model organism to work with. And that's what really struck me. So the sort of balance of doing science for science's sake, but also having another sort of more tangible practicality to the work. So it means that you're easily able to take work which you've done and apply it to other fields which might help them with their research as well. When I was looking for PhDs, I was very much aware that one of the criteria I wanted to have for my PhD was to be working hands-on with insects, working with them on a day-to-day basis, on a practical level rather than just a computational level as well. The wax moth is quite a dull, drab, brown, grey-looking moth. They're quite small. They're about 1.5 centimetres to 1 centimetre. They're from the family of moths known as snout moths because the females have a sort of beaked nose, but really they're just a very dull, drab moth. So the larvae themselves, they start off about 1 millimetre long. They're just a tiny little sort of whitish worm, and they grow up to about 2 centimetres long, and they've got a sort of a brownish head, a set of eye spots, and then it's a sort of white body with 6 true legs, and then 8. They're known as pseudopoda. They look a bit like legs, but they're not actually legs. They're sort of fluid-filled sacs with some claws on the end. It's actually the larvae themselves which are quite interesting because they parasitise beehives. So the wax moths will lay their eggs on the edge of bees' nests in little crevices, and then when the little eggs hatch, the larvae will crawl out and they'll invade the bee's hive, and it will start to eat all the honey, all the wax, and all the old skins that the bee larvae have molted. The parasitism of bees is it's quite an opportunistic one because beehives are full of very nutritious substances. Honey is basically pure sugar almost. The wax comb provides fats, it provides proteins, it provides lots of nutrients. The molted skins from the bees' larvae are actually quite nutritious as well. So it makes sense for something to maybe try parasitize it. Pheromones are quite an important part in all insects' life normally. They're used as sort of signalling molecules to let particular insects know at what time of their life they should be doing something. It's used in a way to let a male let a female know that he wants to mate, he's of the same species, and they produce these compounds, and it results in a very strong innate response from the target's organism. So the females, she'll lay her eggs in crevices nearby the bee's nest, and when they hatch, the little larvae will actually crawl into the beehive, they burrow in, and then they start releasing their own semiochemicals. And these semiochemicals will actually cause the early larvae to aggregate together. What that means is that when they're together, they can break down the wax comb more easily. It benefits them to be together when they're very small because they'll struggle to break down these big bits of wax comb because their mouthparts are very small, whereas if there's lots of them together, they can break it down more easily. And then later on in their life, as they begin to get a bit bigger and they begin to get to the stage where they start to turn into adults, and in that process is known as pupation, when they undergo this metamorphosis from a sort of worm-like larvae into a moth shape. Because they want to all hatch at the same time so they can all mate at the same time, they have this aggregation pheromone again, which encourages them to be attracted towards other pupating larvae. So they'll all tend to migrate towards the outside of the wax comb together. They'll pupate together, and then they all emerge at a similar time as well.
Wax Moths 1. Galleria melanella. When we were waxworms, we sought the interstices, pushed the soft and boneless concertinas of our bodies into hidden places, found the honeyed darkness, its nectar-dripping walls, made ourselves at home in the honeycombs, shedding 7 skins, tunnelling under the capped cells, pale flesh in the honey stacks, pale flesh voluptuous in the living library of grubs, fingers demanding jewels, growing plump, gluttonous for royal jelly, honey, wax, larval skins of bees, our bodies glistening and sticky, an orgy of naked eating, as the bee kind scurry to provide, to tidy, to remove the cell caps we've dislodged. Bald brood, the bee grubs dream unskinned, their raw, unready gleam as we consume, track to the edge of the hive. If the products don't suffice, we eat the larval bees, then sleep in hanging dorms of shelled cocoons, a dangerous, singing mobile in your daughter's room. We wake, we split, disperse, get out fast like a bat out of the moon, pursued by a comet tail of bees come to, fallen from enchantment, suddenly alert to the grey spoils of their ravaged home, its empty combs, a mess of frass and filthy webbing, the nursery's voided multiplex, its blackened cells, the parasites revealed, backing out, turning tail, fat colonials fleeing with their spoils on grey and graceless, homicidal wings.
My research has really formed in 2 parts, looking at cell division, or the process of mitosis, so the equal duplication of cellular material and splitting into 2 daughter cells, and looking at that in Lepidoptera, or moths, and butterflies. So normally when I'm looking at cell division, I'll be looking at the embryonic stage, so the egg stage. If I want to collect cells, you can mash them up, but normally what we'd do is we'd actually look at the cells as a whole. So within the first sort of 6 hours, they go from being 1 cell to being about 1,000 cells, but these 1,000 cells are all 1 layer of cell, so they basically form a sort of coating, imagine, around the edge of the egg, 1 cell layer thick. So it's actually a very good way to look at cell division, because you don't have all the out of focus stuff from embryos behind it. My project started with taking a whole load of proteins which are known to be important in Drosophila melanogaster, which is fruit flies, and taking antibodies raised against those proteins and seeing whether they localize in moths in the same way. And this was a spectacularly unsuccessful experiment, because essentially none of the antibodies worked against the moth proteins, because the moths and the flies have diverged enough that the shapes of the proteins mean that the antibodies can't recognize them both. So they work against Drosophila proteins, but the shapes of the moth proteins are too different. So what we've been doing instead is taking purified proteins and labeling them with a fluorescent dye. So this is a dye that will glow up when you shine a light of a particular wavelength of it. And we take the early embryos and we chemically remove their eggshells using a bleach solution. And then I take essentially like a very, very, very thin needle, and I inject these fluorescently labeled proteins into the embryo. And what then happens is that these proteins are incorporated into the dividing cells, and you can actually see the sort of kinetics, localizations, and timings of how these proteins move and are drawn to certain bits of the cell during the cell division. The second bit is, so the microbiological community use these moths as model organisms for infection. So you can take a nasty pathogen which you want to study, and instead of injecting it into mice and rats to see how pathogenic it is and how much you need to kill something, you can use it in waxworms. So it's a good way to save the usage of mice and rats and reduce the amount you use. But one sort of limitation so far is that they don't have any mechanism to create genetic tools to create genetically modified versions of these moths, which and the use of transgenics or these genetically modified versions is very important because it's useful to be able to knock out particular genes you're interested in, or to put in other genes, or to fluorescently tag proteins that you want to look at inside the organism. So the other part of my project has actually been to create transgenic moths using a variety of techniques. It's quite a time-consuming process because what we've tried to do is put genes into the moth genome which encode these fluorescent proteins again. And it's something which I've been working on for about 2 years, but hadn't had an awful lot of success just because we could see that they were incorporating in the original generation, but they weren't going on to the next generation, which is important. But as of about a month ago, I've had my first transgenic, so we've created the first transgenic strain of this species of moth in the world, which is quite cool. So a transgenic wax moth produces a protein called green fluorescent protein, and this is being driven by a sort of upstream bit of this gene, which means it's produced in every single cell in the body pretty much. So the entire body glows bright green whenever you shine a particular wavelength of light on it. And then we've also put a second fluorescent protein on because we wanted to have 2 markers to screen, and this one is it's one which localizes specifically to the eyes and the neurons around the eyes and the brain. So particularly when the larvae were smaller, if you shine a different wavelength, but a particular wavelength of light on it, the entire brain, the optic neurons, and the eyes glow bright red. So it's got bright red eyes and brain and a bright green body.